Hello, everybody, and this is the virtual lecture on soil-borne and arthropod-borne bacterial diseases. And you'll notice in this chapter, like the other chapters, that the symptoms are going to kind of be similar within this group of bacteria that are the soil-borne and arthropod-borne. In a similar way that the upper respiratory tract infections maybe had symptoms, say, of cough, persistent cough, dry cough, and then foodborne illnesses were more associated with the diarrhea, nausea, and then but you could like differentiate between the different bacterial diseases by the subtle symptoms, say blood in the stool or blood in the sputum or something along those lines. And so keep that in mind as we're going through this chapter. And so soil-borne bacterial diseases. And so the main thing to remember is that these things develop from endospores. And so and if you think about bacteria living in the soil, that soil could be dry, it could be moist, and these things want to live inside of something. And so if they're in the soil, they're probably going to produce a spore. And so you could kind of remember soil, spore. Those kind of go hand in hand. And if you remember, an endospore is the same thing as a spore. And so the first one on the list is anthrax, and this is a zoonotic disease. Zoonotic means it can go between animals and humans. It's a zoonotic disease of large herbivores that ingest the spores while grazing. And so anthrax is the disease. It's caused by Bacillus anthracis. It produces a capsule, and it produces 3 exotoxins. Remember, the bacteria rarely kills the cell. It's the exotoxins that's doing the damage. There's 4 forms of this. There's a skin form, an inhalation form, intestinal, and then there's one that can be injected. And so a little bit more about this Bacillus anthracis. It's a bacilli shaped, and so it'd be a rod shaped. It's gram-positive, so it would have that positive purple protein bacteria. It's aerobic, and this was the guy who identified it was Robert Koch. And so if you remember the Koch postulates in the 1800s, he came up with those. So he took a blood sample from an infected cow, blood, remember, isolated the bacteria and put them into a mouse, and then the mouse came down with the same disease. And so that's the Koch postulate right there. The bacteria normally rests as an endospore form in the soil, and it can survive for decades, remember, in this state. Herbivores are often infected while grazing, especially when eating rough, irritant, or spiky vegetation. So this thing lives in the soil, and you can imagine that an herbivore in a desert situation where it's trying to eat the roots, maybe it's picking up more soil, and then if it's rough or an irritant, you can get these microabrasions inside its GI tract or inside its trachea, and so vegetation can cause these small little micro tears, and then that allows the bacterial endospores into the tissue. Once ingested or placed into an open wound, the bacteria begins to multiply like it does because it's happy, and inside the animal or the human, and then it starts producing endospores. It starts producing exotoxins, and so it produces exotoxins and typically kills the host within a few days or weeks. And so what I was going to say about the endospores is the endospores enter that wound, and then they germinate at the site of entry. And then they're spread by circulation, usually to the lymphatic tissues, and that's where these things really take off. And so there's several different types. And so the human forms, we have an inhalation. This used to be called wool sorter's disease because it was picked up, the spores were picked up from people that were sorting wool. Intestinal anthrax came from contaminated and undercooked meats. Cutaneous anthrax came from skin abrasions that came in contact with spore-contaminated animal products. And the key symptom on that is developed a black necrotic sore. And this was also the agent of bioterrorism in the mail that people were sending around in the mail in 2001. Luckily, this can be treated with regular antibiotics like Cipro. But once in the bloodstream, and so that's how you can get in. And so once it's in there, the bacilli release 3 exotoxins. And we can specifically look at that, the 3 exotoxins. The first one is a lethal factor. The second one is edema factor. And the third one is a protective antigen. And by themselves, each one of these things are not toxic, but the combination makes it incredibly lethal to humans. And these 3 toxins, when they're mixed together, are the primary agents of tissue destruction, bleeding, and then death of the host. Even if antibiotics are administered late in this, in the bacteria are eradicated, the host can die of toxemia because the toxins produced by the bacilla remain in the system, at lethal doses. Recent evidence also shows that the bacteria in the disease anthrax targets the endothelial cells that line the serous cavities, such as the pericardial cavity, the pleural cavity, and the peritoneal cavity, lymph vessels, and blood vessels. And so when it gets in there, the exotoxins cause this vascular leakage of fluid and cells, and then ultimately hypovolumetric shock and septic shock. Anthrax also, might be more prevalent where you have, domesticated livestock grazing on an open range where they're mixing with wild animals. And so sometimes these wild animals mixing with domesticated animals can bring that in. Even though it's present everywhere, but these wild animals, they're not going to get treated. If you have a domestic animal, a domestic cow, you're going to treat that thing a lot sooner than you would, say, a wild animal. Many workers who deal with wool and animal hides are also routinely exposed to these spores, but it's usually not sufficient enough to develop anthrax infections. And so a lethal infection is reported to result from inhalation of about 10,000 to 20,000 spores. And so compared to like TB where it's 1. And so the number of that can fluctuate back and forth. But anthrax is kind of a particular nasty one. The next one is tetanus. And tetanus causes hyperactive muscle contractions. Hyperactive muscle contractions. And so this is caused by the bacterium Clostridium tetani. And wounds from these soil-borne contaminants introduce the endospores to tissues. And then once it's in there, it produces a powerful neurotoxin called tetanospasmin that blocks the inhibition of muscle contractions. And so the muscle can contract, but it can't relax. And so you end up with, lockjaw and stuff like that. I always kind of bring up this tetanus associated with rust and rusty nails. And so the rust, of course, doesn't cause the tetanus, but the rusty nail is usually in a environment where the Clostridium tetani can live. And because of rust, if you can imagine a rusty nail, it has a huge surface area because it's not smooth anymore. So there's lots of surface area for spores to reside on. And then once you step on the nail, like I did in 5th grade, I didn't get tetanus though, but the nail goes and can puncture deep into the tissue and then deliver these endospores, There's a perfect little inoculation deep within the body at the site of the wound. And it used to be called lockjaw because of the jaw muscle stiffness. Treated with penicillin, antitoxins. And there's also immunization, and so you can get a tetanus vaccination. This also, interestingly, affects skeletal muscle. And so if you remember the type of skeletal muscle is a type of striated muscle used in voluntary movement. Other muscle types, such as cardiac muscle tissue, heart muscle tissue, cannot be tetanized because of the intrinsic electrical properties. If you think about how a heart beats compared to how a arm muscle contracts, they're two different critters. And so tetanus will keep your heart beating, but it'll make your back do that, like this poor kid. The next one is gas gangrene, and this causes massive tissue damage. And this is by Clostridial parapharynges. And this produces a group of toxins and hydrolytic enzymes. It's also known as Clostridial myonecrosis or just myonecrosis. And so you might see it as either one of those, which is also what dead tissue is. And so dead tissue, muscle necrosis, myonecrosis, dead tissue myonecrosis blocks the blood flow, causing gangrene, and then gas is produced by the bacteria below the skin, causing it to bubble. Pleasant, isn't it? Treatment is debridement, antibiotics, and you can also use a hyperbolic chamber. And this is considered a medical emergency. But gas gangrene is caused by this exotoxin-producing Clostridium species. And there's a couple that can cause it, but the main one is that parapharynges, and it's most commonly found in soil, but it's also found in the normal gut flora of animals. And the exotoxin that's found in parapharynges type A strain, so there's different strains, of course, that's known as the alpha toxin. And so there's different species that can cause it, but parapharynges will have one strain called a type A strain. And they call it a type A strain because it can produce an alpha toxin. The alpha toxin is a lethal toxin, and what that does is that's what causes it to kill the muscle, and then it also increases vascular permeability, and so then all this stuff starts kind of dripping out. And then the environmental bacteria may enter the muscle through a wound, and then once it's in there, kills the muscle, and then the toxins destroy nearby tissue. The bacteria produces gas at the same time. Because if you remember from our last chapter, that metabolic activities usually result in off-gassing of some kind. The next one on the list is leptospirosis. It's an emerging zoonotic disease found worldwide, and this is it's caused by a corkscrew-shaped bacteria called Leptospira, and Leptospira was the one we talked about earlier in this semester. Signs and symptoms can range from none to mild, such as headaches, muscle pains, and fevers, to severe with bleeding from the lungs and meningitis. If you remember meningitis can be caused by a whole host of different things. But once the infection passes through the meninges, then you have meningitis. If the infection causes the person to turn yellow, then that usually means that you have kidney failure and bleeding, and this is Weil's disease. If it causes a lot of bleeding and seeps into the lungs, it's known as severe pulmonary hemorrhagic syndrome. Leptospirosis is transmitted through the urine of infected animals, and it's contagious as long as that urine is still moist. And although it's been detected in reptiles and birds, mainly it's mammals that are able to transmit the bacterium to humans and other animals. And so rats, mice, and moles are important primary hosts. And this thing can be treated pretty simply with antibiotics. And it also has prevention here. And so you can take a mild dose of, like a prophylactic dose of antibiotics, some of the cyclines, and you can kind of prevent it from happening. And so here we have plague. So plague this is the terrible one. This killed an estimated 25 million people in Europe. It is like just gnarlyville. It's carried in rodents and spread by their fleas. And it's the plague is the disease, and it's caused by the bacteria agent Yersinia pestis. And depending on the infection and the sanitary conditions, it's not only spread by that, but it can be spread by the air, direct contact, rarely by undercooked food, but usually it is by fleas. The symptoms depend on the area of infection. So you can have a bubonic plague in lymph nodes, you can have septicemic plague in blood vessels, pneumonic plague in lungs. It's treatable if detected early, and it's still relatively common in some remote parts of the world, and also some less remote parts, like the United States. And so it spreads through the lymphatic vessels of the infected human until it reaches a lymph node. And there it stimulates severe hemorrhagic inflammation that causes the lymph nodes to expand. And that's the bubonic plague. And so that's where, one of the key characteristics is a bubonic plague is that the lymph glands get really swollen. And if the lymph node is overwhelmed, the infection then passes into the bloodstream, causes secondary septicemic plague. And then if the lungs are seeded, it can cause a secondary pneumonic plague, like pneumonia. And so, like we said, Yersinia pestis causes 3 forms of the plague. The bubonic plague, where cells accumulate in the bloodstream and localize the lymph glands. And then when that gets swollen, they call them buboes. Septicemic plague spreads through the bloodstream, and this one was then known as Black Death. And then the pneumonic, which is like pneumonia. Pneumonic plague is highly contagious respiratory droplets. And so this little guy is still here. Yersinia pestis is still here, and today it's found in the desert in southwest United States. And so transmission of this to an individual can be caused by many different ways. And so it can be by droplet contact, direct physical contact. It can be sexual, indirect contact, usually touching soil contamination or contaminated surface. Airborne transmission, fecal-oral, vector-borne. An example, 7-year-old girl in Colorado. She was diagnosed with Black Death. She thought that she had the flu. She felt sick after camping in southwest Colorado. And she came in to the doctor's office with a 107-degree fever. And the primary care physician didn't pick up what was going on. Maybe her symptoms hadn't gotten as far. But then when they took her to Denver, they found that her lymph glands in her legs were swollen and very, very tender to the touch. And they identified it, and then they treated her with antibiotics, and she made it. She's okay now. And there's gopher colonies in Utah where they've identified a bunch of bubonic plague um, um, dead animals. In that same one, there was a kid that was living in Phoenix, I think, and he went hiking and he came back. He was an athlete, and then came back, felt sick, locked himself in his dorm room, didn't feel well. 2 days later, he was dead. Bubonic plague. So the stuff still exists out there, and that's why those life histories of where people have been is an important thing to discuss. And the next one is uh, tularemia. Tularemia. Tularemia has more than one disease presentation, and the infections can involve extremely small infectious dose causing rabbit fever, or it can go all the way up into more severe stuff where it infects the lymph nodes. There's really two types of this. There's a type A and a type B. The type A is found in rabbits, hares, and pikas in North America. These are the marmots, hares, and rabbits. And it's highly contagious in humans and in domesticated rabbits, this type A. Type B, which is a subspecies, kind of like a serotype, that occurs mainly in aquatic rodents such as beavers and muskrats. And then this one also was present in hares and small rodents in northern Eurasia, but you don't need to know that. But it's less virulent. In January 2011, here's kind of a little bring it home. Researchers searching for brucellosis in feral hog populations in Texas found this little guy in 2 counties. And so, me being from Texas, and people in Texas shoot these feral pigs quite often because they're kind of an invasive species. And in two counties, they found tularemia in these feral hogs, and so there's a little bit of concern because they haven't found these things in feral hogs or pigs, and so it seems like the species has kind of jumped over to pigs, and so even my brother-in-law, he shoots these things all the time. There's a concern that because these pigs cruise around so much that there's a possibility that you could have a zoonotic problem in some of these counties in Texas and the surrounding ones, and so could be present. Next one is Lyme disease. Lyme disease it really can be divided into a bunch of different stages, and you can read the stages on the slide here. And then the spring, the eggs, lays your eggs after dropping them from an animal host. And then early summer, the eggs hatch, and then you have these little baby ticks, and then the ticks get onto a deer, like a deer mouse, and then they can infect both the deer mouse and, because they're transmitting through the tick, then they can grow into large size adults, which then the way that they present themselves as the adult ticks is they hang out on these branches and literally like jump onto the person as they walk past you or the dog. And this is caused by a bacteria, Borrelia burgdorferi, is transmitted by the bite of a small tick. And so there's these ticks. You have different size ticks, and so if you guys have ever seen ticks before, they can be kind of nasty. And the way ticks operate is they stick their head down into the body and then suck the blood, and then they swell up with blood. They're pretty nasty looking. And so there's several different tick-borne diseases And they're listed here: Rocky Mountain spotted fever, the one we just talked about, Q fever, Lyme disease, and southern tick-associated rash illness. But the stages, and so if you had a tick bite, somebody came into the ER or whatever, and they had a bullseye rash, beginning at where that happened, flu-like fever, aches and pains. If untreated, it can spread to the skin, heart, nervous system, and the joints. Late stage develops chronic arthritis and swelling in the joints. It's treatable by antibiotics in early stages. And what they found is that early treatment is the key to this thing, because if it goes a little later, you can encounter problems. And this can cause problems because if you remember in one of the earlier lectures, that bacteria cells, when they're stressed out, they can form an endospore, or they can they develop a persister cell, which is something that can hang out for a long amount of time so it kind of has a protective sheath around it, and it can stay in that state for a long time. And that's what Lyme disease typically does. And so we have let's see what the next one was. And so what happens with Lyme disease is you have these persister cells. You treat it with antibiotics, and you're killing the live, active bacteria cells. But what happens is then the persister cells, then they come back later. And so there's cases of this where these persister cells then come back, and you're essentially reinfected. It's the same thing as having another tick bite. And this explains why the persister cells can evade antibiotics, and then they come back a little bit later on. And so it's pretty difficult to treat these things, especially as it's gone on. Relapsing fever is carried by ticks and lice. This is tick-borne relapsing fever is caused by same genus, but it's a different species of bacteria. And on these little guys, fevers can reoccur as many as 13 times as these spirochetes increase in number. What's pretty interesting about these little guys is there's no vaccine available. And because these spirochetes, they go through an antigenic variation. So there's a term for you that I promise I'll ask on the next test. Antigenic variation. And so the pathogen, what it does is it changes its surface proteins. And so the surface proteins, lipoproteins, the major proteins on the outside, have only 30 to 70% of their amino acid sequences in common. If you remember, proteins are made from amino acids, and so only 30 to 70% have them in common. And so they kind of develop a new face, a new identity. And then antibodies in the blood that are binding and clearing these little guys, they don't recognize the new faces. And so it's kind of a double whammy. You can't do the vaccine, and then your body has trouble identifying which ones are the potential dangerous ones. And so this antigenic variation is common along pathogenic organisms. And so we get into other ones, but that's a pretty interesting little guy. And so here we have some more arthropod-borne diseases. And so the next group of diseases, rickettsia and urechral diseases. These are arthropod-borne diseases. Now, the thing about both of these types of diseases are these are obligate, so they have to. They've got to go to the party. Intracellular parasites, except these are bacteria. And so bacteria can also be intracellular parasites. So these are obligated to live inside a cell. And so, but these next set of slides are these two types of diseases. And they're transmitted usually by lice, fleas, ticks. And they're intraobligate parasites. And pretty much these infections, they involve a characteristic rash. That's kind of the key symptom there. And so Rocky Mountain spotted fever, rickettsia, is transmitted by hard ticks. Symptoms are high fever, headache, skin rash. What happens pretty much with these guys too, there's about a 5%, 3 to 5% mortality if untreated. And there's a picture, you can see these little intraobligate parasites there on that slide on the left, and then you can see the rash on the right. But what happens with these guys is they get in there, and then they start breaking apart and killing the cells. And when that happens, you can imagine if they're doing that in these small vessels, and so you start to bleed out, and that's what causes these rashes. And so a rash, if you get a hickey, you're disrupting the blood flow in these capillaries, and that's what's happening. You're causing these capillaries to bleed out. Next one is epidemic typhus. This is not typhoid fever, this is typhus, and it's transmitted in epidemics. As many as 75% can die. High fever, rash. It's associated with famine and poverty. This is pretty much transmitted by body lice. I'm not going to ask a whole bunch of these. Like on the test, I want you to know that on these soil-borne and arthropod-borne diseases and pathogens, that some of them are kind of standalone and some are intercellular obligate parasites, bacteria, because bacteria can be a parasite. And examples of those would be Rocky Mountain spotted fever and typhus, as well as murine typhus, scrub typhus. And so any of these rash-borne diseases, maybe that would be a better name for it, is the rash-borne diseases. So these diseases that are arthropod-borne, a lot of them, especially there are intracellular obligate parasites. And then there's some new ones that are also emerging in the U.S. and but we're really not going to get into those. But really, what it is with new metrics to be able to identify different bacteria species. Of course, you're going to find new emergence, and so has the disease persisted forever? Probably. Are techniques of finding them better? Yes, and so therefore you're going to see a greater amount of them. So and so you can have emerging diseases, or you can just have better sensitivity to measuring them. So hope this works out, and look forward to seeing you guys in a couple days. I'm in Boston. Adios.